So I want to run through a series of screencasts to make sure that you've gained from this unit the things that I wanted you to gain from this unit. You've been reading a bunch of stuff in your book, but there's some stuff that's not covered particularly well in your textbook, and I wanted to make sure that you had that information. So this is going to be a series of screencasts that I want you to look at this week as you reflect on the COVID-19 pandemic, which is what this set of 3 weeks of material has been about. So by the end of this unit, you should know all of these things, including how vaccines differ from one another and how the human body mounts an immune response to diseases and how diseases go about the process of infecting us. This involves things about blood cells and how your immune system is tied to the different types of blood cells. And it also gets into some cell biology, looking at what exactly it is that mounts and causes the body to mount an immune response. So if you think about the history of disease in the United States, it basically goes all the way back to the first colonists who came to the United States with smallpox outbreaks in New England in the 1630s. These smallpox outbreaks affected the Europeans, but they also affected Native Americans. The Narragansett Indians lost 90% of their population. The Iroquois, which was a group that was only estimated to be about 160,000 strong, by the end of the 1633 outbreak, they had lost 140,000 of the 160,000 Iroquois who were around at the time. Smallpox crops back up in 1721. 6,000 out of 11,000 people who lived in Boston died as a result of that. Eventually, in 1770, we had a smallpox vaccine developed, which then began to reduce the prevalence of smallpox. Note the large amount of time between the occurrences of smallpox and the development of a vaccine. We'll talk more about the development of the smallpox vaccine later on, but this was the first instance of a scientifically proven vaccine that worked. We have yellow fever happening in Philadelphia, and we didn't get a vaccine developed until 1953, even though the yellow fever outbreak in Philadelphia occurred in 1793. We have cholera in New York in the mid part of the 1800s. We have scarlet fever once again, middle of the 1800s. We have typhoid. This is the Typhoid Mary outbreak in 1906 in New York. And one of the things that you'll notice is that in many cases, these outbreaks are limited to a small geographic area. An outbreak in New York, an outbreak in Philadelphia, etc. And they happen to be large population centers. And this is true of vaccines, true of diseases. Diseases do better where there are lots of people. And back before you had good abilities of people to travel, disease would outbreak in a particular place and pretty much stay there because people were not as mobile in the past as they are today. As we begin to move into the 20th century further, we have the development of a typhoid vaccine. This is a toxoid vaccine, a vaccine that is developed against the toxin that is caused by a disease-causing organism. And you'll notice that I'm putting instances of outbreaks as lines on this timeline, and I'm putting the development of vaccines as these broad arrow points so that you can see when outbreaks occur versus when vaccines are developed. We have the global flu pandemic of 1918, sometimes referred to as the Spanish flu, although we generally don't refer to that in science. This prompted the United States to want to develop flu vaccines, but they took a while to develop. We have diphtheria in 1921 to 1925, followed immediately by the development of a diphtheria vaccine. We have polio, which was prevalent in the United States up until 1955, at which point we developed polio vaccine, which caused a dramatic decline in polio. We have another flu pandemic in 1957. Once again, killed over 1 million people globally. We have the AIDS epidemic of the late 1970s and 1980s. We still do not have a vaccine for that. We have a recurrence of measles in the 1980s and early 90s, even though a measles vaccine had been developed back in 1963. We'll discuss in a minute why that disconnect might have occurred. And then we have H1N1 flu in 2009. And then, as you all know, we have COVID-19. And basically, COVID-19 occurred, and the vaccine for COVID-19 was very quick after that happened. In spite of the quick development, still 1.2 million Americans died in that particular pandemic. So one of the things I want you to look at as you look across this timeline is, in some cases, very long periods of time between the occurrence of disease and the development of vaccines. In other cases, the vaccine followed very quickly after the epidemic of a particular disease. And in other cases, we had a vaccine for measles, yet we still continue to have outbreaks into the 1980s. So if you think about this, you can think about diphtheria, any of these diseases, and see the prevalence of the disease and when it began to decline. Pay attention to the fact that the y-axis varies a lot. Measles outbreaks are much bigger than diphtheria outbreaks and rubella outbreaks. But I want you to pay attention to the timeline of when vaccines were developed. So we have a vaccine for diphtheria developed in the 1920s, and then we have a decline in diphtheria that doesn't occur really until the 1950s. Measles, we have a vaccine developed in 1963, and it declines very quickly after that. But then you see these little outbreaks that occur once again all the way up into the 1980s and 90s. These actually turn out to be outbreaks that were due to the vaccine having an attenuated form of the disease-causing organism that wasn't attenuated enough and ended up giving people the disease. And so you got these isolated outbreaks. Mumps, once again, mid-60s, vaccine was developed and a very quick decline. Pertussis, not so much. And so you see that pertussis whooping cough vaccine was developed very early on, but they didn't really start to see significant declines until the 1940s and into the 1950s, which is possibly due to the fact that with the development of the polio vaccine in 1955, people became more aware of the utility of vaccines, and people might have been more likely to get other types of vaccines that were available. And then we have rubella, once again, a 1960s era vaccine, and you saw a decline in rubella very quickly after that. So what I hope you get out of this historical perspective of vaccines and their effect on the prevalence of the disease in a population, in this case the United States, you would take home the idea here that vaccines actually are effective at preventing the spread of diseases. So if you think about vaccines, and we look at measles in particular, you see that in 1963, the vaccine was licensed. You saw a significant decline, and you got a series of outbreaks after that. Once again, these were mainly due to poor controls on vaccine production more than anything else. But when you look at this timeline more recently, starting in March of 2023, going up to the present, essentially, you see that in the year 2025, you have a relatively large outbreak, although it's not in the hundreds of thousands, it's in the hundreds of cases. And so you might be thinking to yourself, well, what is going on where we're having outbreaks of measles after measles had been declared eliminated in the United States? So in 2000, we declared measles just a done thing in the United States. Well, when you look at measles outbreaks, you find that they are concentrated geographically in one particular place, the state of Texas and the adjoining state of New Mexico. And when we look at the percent vaccinated, we see that Texas happens to be a state that is less than 95% vaccinated, and 95% is generally the level of vaccination that epidemiologists think is the level that you need to be at in order for herd immunity to take effect, so that there are enough people vaccinated that a disease will tend to not spread. So let's ask ourselves, and we're not doing great in Oklahoma here, so let's ask ourselves what's going on in Texas. So we can zoom in on Texas a little more closely, and we actually see that the measles cases that are present are located in a relatively small geographic area in the western panhandle of Texas. And when we zoom in on this area, we can see that the largest cases happen to be in these counties where levels of vaccination are relatively low. And as soon as the disease starts spreading out and comes into contact with counties where the level of vaccination is above 95%, you begin to see that the spread of the disease essentially gets stopped. So once again, the take-home point of this is that herd immunity actually works. If you live in a county where vaccination rates are relatively low, the disease, if it shows up, can then spread relatively quickly, and it will spread until it comes into contact with a population or a set of populations that are relatively well vaccinated, at which point the disease can no longer spread. So when we think about diseases, we need to think about the structure of a cell. Cells are surrounded by a plasma membrane. This is a plasma membrane. It's made up of a phospholipid bilayer. This phospholipid bilayer is embedded with proteins, and these proteins have different functions, and we'll talk about those different functions later. But these functions primarily serve to allow the organism to recognize its own cells, and it also uses this information to recognize other cells and mount immune responses. And so when we think about disease, one of the important groups of cells that we have to think about are blood cells, because that is where our immune system is generated from. All of these blood cells have plasma membranes, and these plasma membranes are embedded with these membrane proteins, and there are different kinds of cells that are involved in different kinds of immunity. So there's what we refer to as innate immunity, a generalized immunity that isn't specific to any particular pathogen. This is sometimes referred to as the general inflammation response. And then there's what we refer to as adaptive immunity. An adaptive immunity is an immunity that occurs in response to a particular epidemiological challenge. There's an antigen that is located in the outer coverings of disease-causing organisms, whether that's a capsid of a virus or if it's a cell membrane of something like a bacterium, and your body mounts a specific response to that specific pathogen because of markers that are on those cells. So when we think of innate immunity, we can think of physical barriers like your skin, your respiratory tract, your stomach. Your stomach is acidic. It gets rid of a lot of pathogens just by killing them with acid. And then there's chemical signaling. Chemical signaling brings about changes in cells and tissues that serve to limit the infection to a particular area of the body and also recruit cells to the infected area. Like I said before, this is the general inflammatory response. Adaptive immunity, on the other hand, involves blood cells interacting with a particular type of pathogen, and so it mounts a specific immune response to that pathogen. Blood cells then produce antibodies to the antigens that are found on the plasma membrane or the capsule of the pathogens, and specific cells will be produced that target a single or a small family of pathogens. And this is a type of immunity that lasts for long periods of time because your body also creates what are called memory cells, cells that remember this pathogen and allow you to mount a very quick immune response if you come into contact with that pathogen again in the future. Vaccinations give your immune system exposure to the markers that are on pathogens so that it stimulates an immune response in your body, so that once again, if you ever do come into contact with that pathogen for real, your immune system already has experience with it and you can mount an immune response very quickly. So the way we do this is by giving you a vaccine. Vaccine is just a substance that is designed to stimulate a response that gives you then this adaptive immunity. Usually it's been formed in the past from an attenuated form of the pathogen. This is either a weakened or a dead form of the pathogen. And It's actually the proteins and glycoproteins on the outside of that organism that serve as antigens that stimulate this immune response. And in some cases, more recently, we have developed messenger RNA vaccines that do not require a whole organism to be injected into you. You just get a little section of RNA, and then your own body produces the antigen that then prompts an immune response. So it's the genetic code from the antigen that triggers the immune response for you. So that's a brief introduction to disease and vaccines in a very superficial sort of way. The next screencast will be about cells and how they're structured, and then thinking about blood cells in particular. So I will see you then.